In this video, we are going to just review the basics of pathway analysis and the ideas behind it. So there are two ways that we can collect data to run pathway analysis. The first and foremost, we need to have experiments and data from them that is confirmed through statistics. Usually that is a very long process and a multi-step process where multiple investigators look at the same question in multiple model systems, in multiple settings, and they get a whole lot of data, each one building upon the work of the other, so that they can get a host of different genes and proteins that may be affected in response to their treatments or the disease or whatever the question is that they've been looking at. Once a lot of experiments have been done and a lot of data has been collected, then that data can be further combined to run pathway analysis. Another way that you can use experiments to collect a large amount of data is through gene expression arrays or protein arrays, where we look at the RNA or proteins that are present in a particular condition or a cell, and you compare and contrast different treatments, different time points, or different samples, and run pathway analysis on them to see which pathways, which bigger systems are changing in response to those conditions. A different way that we can approach this is through a literature and knowledge-based research, where we take data that is already published from experiments from before and don't run the actual experiments ourselves, and take the knowledge that is contained within them to make a list of genes and proteins that we can analyze in response to our conditions. Using pathway analysis, we can then gain further insight into that data and synthesize those conclusions in a different way. Here, this is a schematic showing the same information in a different way, that we mine information either through data-driven objective, where we do a list of experiments and do gene expression or microarray analyses, and then extract the pathway information from them using different types of platforms available to us, whether they are paid platforms like Ingenuity or specific pathways. Information can be gotten through several open source programs like Reactome, like the one we'll be using today. Using that information, we can then assemble the pathways and add our annotations into a pathway draft, which is a basic idea on what we think is happening in our particular condition in response to our treatments or times or conditions. And then we refine that pathway in a kind of a cycle of this, where we take our pathways information as we know it, run more experiments to validate those findings, and then update our pathways as new information becomes available. So why we would run pathway analysis is the biggest reason for that is that we want to get a kind of a broad idea about what systems are getting affected at a macro level in an organism or in a cell in response to a certain condition. We also would want to glean a large amount of data in the case of high throughput experiments, where you get lots and lots of data on all the genes that are present inside a cell under various conditions, or all the RNA or proteins that are expressed in a particular cell in response to certain conditions. And we are gleaning through all that humongous amount of data to look for important information, to look for things that are changing, to look for things that are important for the biological pathway or question that we are trying to answer. The high throughput experiments themselves do not produce biological findings. However, they do provide you with ideas on which pathways or which areas are of interest, which areas are most affected by the conditions that we are looking at and should be studied further in order to find actual biological findings. what this data allows us to do is interpret that data in the context of some biological processes, pathways, and networks that we can then use to develop a hypothesis that can be actually experimentally looked at and validated. So once we get our pathway analysis finished, the few things that we can get out of that depends on the question we were trying to answer. If we had a big gene expression array or a proteomics array that we were looking at, then this kind of pathway analysis will allow us to identify genes and proteins that are associated with that particular condition or specific disease or a treatment. If we were looking at, if our question was to look at a particular condition with and without a particular drug treatment, then it is going to help us find the mechanism of action for that drug, and it is going to help us predict targets for that particular drug. So it will help us predict targets as well as understand how to target the therapeutics. It can also help us integrate a vast amount of valid data, available data from other experiments done by multiple investigators in multiple model systems regarding a topic, into a cohesive integrated system where you can look at how all those experiments have some common nodes and some common genes or proteins that keep popping up. And it can also help us assign functions to genes with unknown functions from before. So if we have a new protein that popped up that we don't know anything about, but when you start to build your pathway and run these analyses, it always shows up in a cell cycle category or in an apoptosis category, then you know that it may have a function associated within that pathway. So the reason why we use pathway analysis softwares instead of looking at them ourselves manually is, one, given the vast amount of data that we are usually using for these type of analyses, it would be impossible to do it without the help of the algorithms and the system built around the softwares that we use. They can be used as a learning tool when we study a group of gene products or proteins or microRNAs. They can also be used as data analysis tool where we use them to look for affected pathways or look for diseases that have similar biomarkers popping up. And finally, the biggest reason people use them nowadays is to use them as a hypothesis generation tool. By using all the information that they already have available in the pathway analysis, they can use that to generate ideas on what effects a gene knock-in or knockout might have. They can affect possible side effects of drugs based on what networks and what big macro systems are getting affected in response to your drug treatment. They can help us provide insight into mechanism of regulation for genes of interest. And it can also help us likely give us, some predictive ability to predict what can happen as a result of our observed changes. Now, within a pathway or in a pathway analysis software, the three parts that we need to understand of are node, pathway, and network. So we should know the difference between them. This is essentially going from looking at a single gene, mRNA, or protein of interest, which would be the node, and it is the one-dimensional representation of your data. So these are the things, the individual genes and proteins and mRNAs or microRNAs that you find, that you input into the software, to the pathways that are associated with those nodes. And then finally looking at the larger networks, which are the bigger cellular functions or regulations that are involved when looking at these nodes and the pathways associated with them. So network is your multidimensional representation of the data, while node is the one-dimensional single gene effect or single protein effect. Once we've run these pathways, there are certain questions we can answer, and those are dependent upon what type of analysis or what type of questions we were answering before that. So depending on the type of data we had and the type of questions we asked with the pathway analysis, we can look at the new things we have learned by incorporating all that data that we input into the pathways analysis software. We can think about what type of things should we expect to learn in general in response to those results. Many times it will help us design new experiments as a follow-up to validate our findings. And then we can think about how this new knowledge can be used to further our research and answer questions related to those as well. So this is, looking at what things cannot be done with a pathway analysis a little bit, or some caveats to that. Now, not every single gene is going to belong to the database in your pathway analysis software. And not every gene is going to belong to a pathway, period. Some genes do not have functions associated with at this time, and they may not show up in any of your systems and may just show up as a separate thing. You also have to realize that the pathways that are generated are based on statistical probability in the provided data set that you are giving it. They are not a biological certainty. So just because you find a particular gene or protein upregulated now doesn't mean it's always going to be upregulated in those conditions. It may be that when you change the cell line or change the model system, the data set changes and the results are different. The third thing is that the context matters a lot in pathway analysis. So the more clear you are in defining how these proteins and genes are affected in your conditions, the more clear you are in your experimental design, the better data output you will get as a result of the analysis. We have a saying at Mayo when we do these type of work that if we put garbage in, we get garbage out. So we have to be very careful in designing our experiments with appropriate controls and appropriate conditions and appropriate time points in order to get meaningful data out of pathway analysis. Finally, whatever results you get, no matter how robust your experimental design, this is just the beginning. This is just the start of your experiment. Using that data that you find from the pathway analysis, you can design future experiments, and you would still need to prove those findings experimentally for them to have any weight. So that's all we have for this basic introduction. Now we will look into Reactome as a tool to study genes and proteins we find differentially expressed.